Hi, Michelle Glass here. Welcome back to another episode in our Chapter 4, Lecture 4 series. And our topic here are those organelles and structures that make up our endomembrane system. So that prefix endo means inside the cell, and then membrane is talking about like the cell membrane and membrane-bound organelles. Remember, we are going to get into the conversation of our cell membrane structure more in the future, so we have a whole chapter where that topic is explored. So let's look at what consists of the what the endomembrane system consists of. So here we have our complete list. So we see the nuclear envelope, which we've already talked about is that outside structure of the nucleus. Our endoplasmic reticulum, there are two types, so I have both smooth and rough listed there for you. The Golgi Apparatus, lysosomes and peroxisomes, where that some ending means body, so these are bodies of these certain types of molecules and having special functions. Structures called vesicles and vacuoles, and then also our cell membrane structure is going to be like the outer border there of our cell, and all of these other structures in our endomembrane system are very often communicating with that cell membrane. So we're going to be looking at each of these in more detail as we progress through. So here I've drawn an example of a plant cell for you. In our presentation on the genetic control organelles, I showed you more of an animal cell design. I know this is a plant cell because I have a rigid cell wall structure, a rectangular shaped cell, and then I do have my cell membrane inside my cell wall because remember that's universal to all types of cells. Notice I have my nucleus, and then immediately connected to that nuclear envelope, I have my endoplasmic reticulum. And our endoplasmic reticulum, think of it as a maze or network of membranes. Again, what's significant about having these membrane-bound organelles and membranous structures is that it's a specialized environment. So remember, we talked about, like, if we were thinking about this as a house, then this endoplasmic reticulum, this network of membranes, are specialized rooms inside of that house. Now, there are two types of endoplasmic reticulum. We can abbreviate endoplasmic reticulum as ER, so that's nice. We don't have to keep saying that whole phrase. We have what's called the smooth ER, which is shown without the green dots. The smooth ER is going to make a lot of lipids. So they're going to make phospholipids. That's part of our cell membrane. So they're helping to make cell membrane. We're going to see cholesterol, which is also part of our cell membrane. We're going to see lipid hormones being produced here. We're going to see carbohydrates being produced here. So think of your smooth ER as making lipids and carbohydrate molecules. Also, the smooth ER is involved in drug detoxification. So if you're looking at human or animal liver cells, you would expect to see a lot of smooth ER to help remove those toxins and break down things from drugs. That can be prescription or non-prescription drugs. If you are familiar with individuals building up a tolerance to a drug so that initially, a small dose was effective and then you had to gradually increase that dosage in order to have the same effect, that is happening because those cells are adding more smooth ER in order to be able to detoxify the body. Another job is to sequester calcium ions. This term sequester I define as storing and hiding. So we'll see this important if you do take a biology 2 and you look at muscle contraction. Our rough ER I've represented on the side with those green ribosomes attached. Remember, we talked about ribosomes in our genetic organelle video. The ribosomes are either free or attached to the rough ER. So it's called rough because it looks rough because of all these ribosomes attached. Now remember, the ribosome is making the protein. That's where protein production happens, but then it can be released into the rough ER and then modified. So we'll see the rough ER is modifying newly produced ribosomes and then packaging them into structures called vesicles. So little bitty membrane-bound structures in the cell, membrane-bound packages, are called vesicles. Think about it as a little special box. And again, anything that's bound with a membrane has its own specialized environment inside. Remember, environment plays a role in protein structure. So keeping it packaged in these membrane-bound rough ER and then in the membrane-bound vesicle is helping to maintain that correct shape for that protein. We will see that vesicles from the rough ER make their way, so they actually get moved along a cytoskeleton to our next organelle, which is the Golgi apparatus. Our Golgi apparatus is a stack of membranes, stack of membrane pockets you can think of. Oftentimes it's described as a stack of pita bread, which is that little pocket bread, if you've eaten that before. So it is not associated with the nuclear envelope. We're further away, and instead of it being like a maze or network, it's going to be these membrane pockets. We see vesicles coming from the rough ER to the Golgi apparatus. The Golgi apparatus might add additional molecules to that protein so it can modify it. It can package it up into its own new vesicle, and it can then send that vesicle to the cell membrane for export of that molecule. Our next structure to look at is called a lysosome. A lysosome is a specialized vesicle produced by the Golgi apparatus. We will see lysosomes present in animal and protist cells, but not in plant cells. The lysosome is containing what are called lytic enzymes. That term lysis I've introduced to you already means to break. So lytic enzymes are enzymes that will help to break down, in this case, old cells, damaged cells, pathogens, organelles, so pretty large structures. If you've been watching Amoeba Sisters with me, they super love cake, and you'll see that as a theme. So if you're an amoeba, then you're going to take food molecules from your environment into the cell. That food vacuole is going to fuse with a lysosome, and so it's acting sort of as like a stomach to help break down material. So it might be to break down things that are harmful to the body, like a pathogen or old and damaged cells, or it could be helping to break down nutrients for the cell to use. If you're a plant cell, you don't have lysosomes, but you have vacuoles. Now, vacuoles are not unique to plant cells. We do see vacuoles in animal cells. We see vacuoles in protists, but we do see vacuoles having a really important job in plant cells specifically. Plant cells will all have this really big vacuole called a central vacuole, and then there can be other vacuoles throughout. The other thing to note here is that plant cells have vacuoles that are acting like lysosomes in our animal cells. So we see that the vacuole basically is replacing the lysosome. So that's why plant cells don't have a lysosome. The vacuole is going to do lots of things. So it can help to maintain osmolarity. We'll be talking about osmosis in the future and osmolarity in the future, but this is basically regulating the fluid content. So that's letting a freshwater cell live in fresh water without gaining too much water, or letting a ocean fish or ocean cells, I should say, because we're talking about this on the cellular level, live in a salty environment. So you can be adapted for either fresh water or salt with the help of the vacuole maintaining your osmolarity. There can be a digestive function. So again, we're seeing that that's the role of the vacuole in the plant cell, replacing the lysosome. We can see it used for storage. So this can be used for lots of different types of cells, but examples of storage might be proteins. In plants, we see flower pigment is stored in vacuoles. We also see that plants often store poisonous compounds in vacuoles, which is helping to protect the plant because it's poisonous and it tastes bad, then it's reducing predation. When we talk about that central vacuole in the plant cell, we'll see that it's very important for the overall structure of that plant cell. Remember, vacuoles are not limited to plants. We're just seeing them having a pretty important role here. Peroxisomes are another structure. These are similar to lysosomes, but instead of breaking down really big stuff like cells and cell debris, we see here that we're breaking down macromolecules. So peroxisomes are important in breaking down fatty acids and other macromolecules. They're also important in producing the enzyme catalase, which we see breaks down hydrogen peroxide, which is abbreviated H2O2. Hydrogen peroxide is a toxic waste product of cellular respiration. And so this is something that cells have to be able to manage. Okay, so let's kind of get the whole picture here like we did with our genetic organelles. And looking at the whole picture of the endomembranous system working together, we have, remember, in our DNA, that process of transcription happening. We already talked about that in our previous video. So that's where the DNA molecule makes the mRNA. The process is happening in the nucleus. The mRNA can then leave the nucleus and take that recipe for making the protein to the ribosome. The process of translation is the actual production of that protein. That protein is made then at that green dot at the ribosome, but then it's released into the rough endoplasmic reticulum where it can be modified. So maybe carbohydrates are attached. Maybe lipid parts are attached. And as it moves through that network of rough endoplasmic reticulum, it's going to finally be packaged by the rough ER into a vesicle. That vesicle then can be transported to the Golgi apparatus, where it can be modified further, where it can be packaged into a new vesicle, where it can then be transported out of the cell. Okay, so that is our endomembrane system. Stay tuned for more. My nose.